Okay guys, welcome back. We are finally on our last clip for this first module, and we are concentrating on that mitochondrion. That's the last organelle we have to talk about in the cell. And we're kind of rewinding back to objective 30, where we need to address certain things about ATP and energy and cellular respiration. So before we really dive into that and you hear my explanation, my description of this, let's watch this little video, little gummy bear experiment, and I'll put a link to this down in the description as well, but check it out. All right, guys, so this is potassium chlorate, which is very good at pulling electrons away from other molecules. It's like oxygen in that way. And for that reason, we call it an oxidant. Okay? So potassium chlorate, we're just melting it here in the tube. Pretty crazy, right? A lot of energy being given off. Think about that next time you have Gummy Bear, right? All right, so I've done this exercise in front of a group before, in front of high schoolers, and I chose a tube that was too thin. It actually exploded the tube. I mean, I was fine and I had protection on, but it actually broke the tube. It's that powerful, just one gummy bear, right? So imagine that, right? The energy that's in just one lump of sugar, which is basically what gummy bear is, or an M&M, or take your pick. You put this in your body and all that energy is there, okay? It so happens that potassium chlorate in that tube was really good at oxidizing and breaking bonds, in other words, okay? And those covalent bonds that were broken, that were in those sugar molecules in that gummy bear, gave out a terrific amount of energy in the form, in this case, of heat, in the form of light, right? Well, that absolutely, obviously, doesn't happen to us when we eat a gummy bear, right? So something's got to be going on. We know sugar is for a quick use fuel item, right? Carbohydrates are for easily used fuel, right? Energy can be gotten out of them. But this obviously must not happen in such a very quick manner when you eat a gummy bear, okay? So what we need to do is we need to address how it is that we get energy out of our sugars and how we harness that energy. We don't just give it off in lots of heat and light. I mean, gosh, right? If you eat a handful of gummy bears, that would be crazy, right? So let's think about how it is that we get more energy by eating the foods we eat. Let's think about it as a large molecule, right? We bring in certain foods, certain organic compounds that have a lot of bonds holding them together. These atoms in these compounds represent energy, okay? So a large molecule is going to have more bonds than a small molecule, okay? And we can just simplify it and say, okay, a molecule with some part of it we're going to call A is bound to a hydrogen here, and it's bound by a covalent bond, so I have to imagine that there would be sharing of two electrons there, right? So electrons are representing that covalent bond. And if we were to want to make this a smaller molecule, we could break that bond, right? We could take the bond with those electrons and oxidize. Through oxidation, we would break that bond. Now, we've used some other terms like catabolism for cutting something down. Well, this is a form of catabolism, but in chemistry we call that oxidation, okay? And it's more appropriate to use in this description. Now, if there's energy being given off and electrons now are to be had, to be used perhaps in a bond elsewhere, right? It's no longer used in an attachment of hydrogen to this A molecule, but what if there's a B molecule, and what if we use the energy and the electrons to form a new bond elsewhere, right? So we're going to make this B molecule bigger, right? This would be making a small molecule larger, right? And that is reduction, okay? Now there are a couple ways of remembering these that are kind of cute ways. Leo the lion says grr, okay? So Leo, so the Leo would be loss of electrons, right? Is oxidation. So electrons were lost over to here. So on this side we have oxidation going on. Gain of electrons is reduction. Okay, and that's what's going on down here. Or you can think oil rig. Oil rig is oxidation is loss. Oxidation is a loss of electrons. And reduction is a gain of electrons. All right, so that might help you to remember these. Now let's just think about, what is A, right? Where I'm just making up things. Let's use a real live compound, something we know about, in place of this AH molecule that we keep talking about. Let's say now we're talking about glucose. And glucose, well, we already know CHO. We already know that about carbohydrates, right? Glucose being a monomer, a monosaccharide, it's going to have carbons, hydrogens, and oxygens. Those are the elements. It just so happens that there are actually 6 carbons, 12 hydrogens, and 6 oxygens in this one molecule. 24 atoms. 24 atoms being held together by lots of different bonds. Lots of energy to come out of that if we break those bonds, correct? Think of it like a candy bar. If I have a candy bar, a big hunk of sugar in front of me, I can start to break that down, right? I can start eating that candy bar, and as I digest that candy bar, I am going to be breaking these bonds and releasing energy. Now, if I release energy the way that we did with that gummy bear, it's all going to be lost, right? So think about the carbons and the oxygens in this glucose molecule. If I do this right in my breakdown of this glucose molecule, I can get carbon dioxides out of that. It's not too far-fetched, right, to think that I could use some carbons and some oxygens and make some carbon dioxides out of this. And in fact, in the process of cellular respiration, this process whereby we oxidize glucose into carbon dioxide, we are going to release that energy. And this is our waste product, right? We breathe that away every time that we exhale. Now, what are we going to do with that energy? We're not going to just release it, right? We want to harness that energy, use it when we want later, right? We want to go for a run in about a half hour. Well, that energy will be there, right? Why? Because we ate food, we had carbs and lots of other food items that have bonds that hold them together, and we then harness that energy. So how do we harness that energy? Well, we can reduce another molecule. We won't call it B, we'll call it ADP, adenine diphosphate. Okay, so it's a diphosphate. It's got 2 phosphates to it. I'm not showing you the actual compound here. Why confuse the issue with a complicated molecular structure of ADP? Suffice it to say, it's kind of like that nucleotide adenine, a monomer of nucleic acids, but it's got 2 phosphates on it. If I use the energy and the electrons that from breaking bonds that hold this glucose to molecule together, I can add a 3rd phosphate, triphosphate, onto this diphosphate molecule. And energy will be holding that new phosphate onto this molecule. Energy I might be able to use later. Okay? So when I oxidize, I break bonds. I'm going to oxidize glucose, get CO2 out of it. What is the important thing I want out of this? I want ATP. I want this molecule that has energy in a bond I can use later. Think of this ATP as just like the original molecule, ADP. Okay. But now I have this special bond that's holding another phosphate on. There's our energy in that special bond, right? That special bond has been made so that we can then break the bond later and use that energy. So let's take this a step further. I can oxidize glucose in the time being. I can make lots of ATPs which have a bond I can break. Now, I have made a bond in the production of ATP. Again, I'll draw it again, right? So we have ADP, and we've formed a special bond with another phosphate. This is the energy I want, okay? This energy that's in this bond holding that phosphate on. All right, so later, what might I want to do? Maybe I want to use the energy in adenosine triphosphate, and I'm going to break that bond. Okay, so I'm going to come along, I'm going to break that bond. That will be then an oxidation again, right? We're going to go backwards with ATP becoming ADP. And the energy from that oxidation we can use to reduce something else. We can make bonds, in other words, someplace else. Where might we do that? Why would we do that? Well, let's say we have a bunch of amino acids in our body and we want to make some proteins out of them. Well, then we need peptide bonds to hold these amino acids together. We need some covalent bonds, called peptide bonds, to hold them together. This is one way to get it. We're going to use energy from ATP and probably an enzyme, because the enzyme will get these amino acids real close to one another, right, to reduce that activation energy, and we're going to get a protein out of it. Now just think of all the other things we might use this energy for to make bonds. What if I want to use energy to put a bunch of glucoses together to make glycogen? What if I want to use energy to put a bunch of nucleotides together to get my DNA? Or sometimes this energy will be used for many of the other types of work that a cell needs to do. Some of the other types of work will allow for movement. So when my muscles actually contract and move my skeleton, well, they're using a lot of ATP. They're using a lot of this energy that's pent up in this special bond. Okay? So we absolutely do need to understand this because energy is really important to the functioning of our cells and to normal functioning of the body as a whole. All right, so I'm not trying to scare you. I just want to show you another way of thinking about the cell. Okay, so now we've zoomed in on a mitochondrion. Okay, so here we have our mitochondrion right here. We've got our outer membrane and we have our inner membrane. Now, we discussed how the mitochondrion is kind of special. It kind of looks like bacteria that exist out there. It's got DNA of its own. And most importantly for our discussion, it's got an ability to give us a lot of energy. And in my mind, when I hear a lot of energy, I think a lot of ATP. Okay? So here they're showing us in 3 blocks a set of reactions that will allow us to take glucose, so here it is, way over on the left side, break the bonds in that glucose, oxidize that glucose in 3 phases. One of those phases occurring outside of the mitochondrion, out in the cytoplasm, not outside the cell, but in the cytoplasm, the cytosol, okay? And then 2 of those phases occurring within the mitochondrion. And there is why the mitochondrion is so important, because to get the most energy out of your sugar molecule, you need the reactions that occur within the mitochondria. Now the question becomes, how much energy are we getting because of that mitochondria? Okay. Now there's a more complex image like this I'm going to show you here, and it shows you more details. Okay, it shows you how much ATP is going to come from each of these stages. And you can go to the page in the book, put in aerobic respiration into the search box in your ebook, and it'll take you to a later chapter. But it'll probably give you a run for your money. It's pretty complex. So we want to break these 2 slides I just showed you down to something just bare bones that you need to know from me for this next exam, okay? And need to understand and remember for later segments of this semester, okay? So let's just break it down. Let's get what we need to know so that we can go away and get this stuff under our belt for the exam this coming Friday, okay? So, in a nutshell, you need to know what's on this slide, these various things about the stages of aerobic respiration. And these are basically going to answer objective 30 for you. Now, first let's start with the title. I'm calling this, am I calling it cellular respiration? Am I calling it aerobic? Well, it so happens that we are a very aerobic dependent being, okay? We're not like certain bacteria that can get by without oxygen. Aerobic means needs oxygen, okay? So I could talk about it as cellular respiration because that's what all of our cells do. But I could also say synonymously pretty much aerobic respiration because our cells need oxygen to perform cellular respiration, okay? So, in a nutshell, what do we want to know? First of all, I want to know the names, each of the names of the 3 stages of this particular cellular respiration equation. I want to know the location where each of them take place, where in the cell, okay? I want to know the oxygen requirements for each of these stages, each of these 3 stages, okay? And finally, I want to know the amount of ATP that is produced within each stage. As an overall, you would want to know the equation that you see at the bottom here, okay? Glucose, this is representing glucose, plus the oxygen that we take in by breathing oxygen in, and ADP molecules, they're the smaller molecule we're going to build up, right? These all have to be coming together to give us CO2, this is a waste product, water, and most importantly, ATP. This is what we really want, is all the energy that we can harness in ATP. Once we have ATP, it's like money we made, right? It's like what I'm doing right now. If I'm going to put a little work in up front in order to make money, right? So like what I'm doing right now, explain to you guys cellular respiration, aerobic respiration, I'm going to get a paycheck, right? I'm going to go over this stuff with you guys. I'm going to get paid for my job, and then I'm going to turn around and I have now currency, right? I have money. I have ATP. I spent a little bit of energy up front, a little bit of time spent there to get ATP. When I need my car fixed, okay, or put a new roof on my house, I'm going to turn around and I'm going to pay somebody, right? I'm going to take that ATP, I'm going to take the energy that I've harnessed in the form of money, right? ATP is like currency for energy. And I'm going to turn around and I'm going to give somebody else that currency. And it's going to get work done for me, right? All right, so let's go back to a simple diagram like this one. It's simpler than it could be, believe me. And when you go to the later chapter in the book, you look this up, your mind could be blown. So let's just answer the questions on that objective 30. What's the overall name? Aerobic respiration. We already did that. What are the 3 phases? Phase number 1, glycolysis. We're lysing glucose. We're breaking it down. What's that mean? Well, we're just breaking the bonds in sugars, right? Think of it like a candy bar. I'm breaking it down. I'm going to eat half the candy bar by the end of glycolysis. By the end of glycolysis, I will have broken glucose in 2. Just so happens the 2 pieces of candy bar are called pyruvic acid. Pyruvic acid would be each of those would make up half of a glucose. The second stage, where I'm oxidizing this glucose, breaking this glucose down, is the Krebs cycle. Now this has to occur in the mitochondria. And electron transport chain, or electron transport system, you could say, occurs also in the mitochondria. So we've got our 3 names: glycolysis, Krebs cycle, and electron transport chain. By the end of glycolysis, we've half broken down our glucose, like a half-eaten candy bar. But by the end of the Krebs cycle and electron transport chain, we will have gotten as much energy as we can out of the candy bar, broken as many bonds as we can. So the names are down, right? Glycolysis, Krebs cycle, electron transport. Next thing, we want to know where they occur. Okay, so it happens that glycolysis occurs in the cytosol. It occurs in the cytoplasm, the liquid part outside the organelles. So, cytosol. The other 2 occur within the mitochondrion. Can't get those done without the mitochondrion. Okay, so we got our second set down. The names and where they occur. The third set of information we need to know about these will be oxygen requirements. Do we need oxygen or not to make each of these stages run? The answer for glycolysis is no. No oxygen is necessary. That doesn't mean that oxygen is inhibitory or stops the process. Oxygen can be present and glycolysis will occur. But oxygen can also be absent and glycolysis will occur. Okay? But when it comes down to the Krebs cycle and electron transport chain, yes, we need oxygen. And without oxygen present in sufficient quantities, these stages will not occur very well at all. Okay? So whatever ATP you would get from Krebs cycle and electron transport chain, down the drains. We don't get a lot of that. Now, our fourth set. We need to know how many ATP, what kind of energy do we get from each of these stages. Okay? So let's write them down here. Glycolysis, well, it's just part broken down candy bar, right? I've just half broken down this candy bar. Half broken down glucose gives us 2 ATPs. A pittance, not very much at all. Once we get into the mitochondrion, however, we start really picking it up. We get 2 more ATPs, again, minimal, from the Krebs cycle. But the electron transport system, 34. We're in the 30s now. If we have a mitochondria, if we have oxygen present so that these can run, and electron transport change going for us is going to get us 34 ATPs. Now, if you add these numbers up and compare them to the equation we just discussed a little bit ago, I said something about 36 ATPs, a nice payload, right? A nice paycheck for doing this. Well, 2 plus 2 plus 34 does not give us 36, right? The reason for this is to transport the half-eaten candy bars, the pyruvic acids, these smaller molecules that result from half breaking down glucose. into the mitochondria. The toll for doing that is 1 ATP molecule per pyruvic acid. So 2 ATPs are lost going in to that mitochondria. And this is why we see 36 when we use our summary on our equation. Okay? So if we again look at this equation, we see that 36, and this is good news, right? We're going to be able to do a lot of energy, a lot of work, build molecules, move our muscles, right? These are all really essential for our survival. Now, this will really, I think, address what you need to know for Objective 30. And I think we've gone over pretty much everything in our objectives. And if you have trouble, remember SI is available, and I've done some hours today. I'll be doing some hours tomorrow on Sunday. And let me know if you have any questions.